So I'm going to tell you today about some research we've been doing in my lab here at NYU for about 10 years. What we're trying to do is we're trying to take tiny little particles and put them together. Sounds simple. So the tiny little particles are called colloids. Colloid comes from the word glue because these things are sticky. Now the thing I want you to notice here on this view graph is what the size of these colloids are. They're typically about 1/100th of the diameter of a human hair or smaller. They consist of lots of molecules, so they're not molecules, they're little tiny particles. And the question is, how do you assemble these groups of tiny particles into useful structures? So we have here's a few examples of colloids that I put together. Paint is actually a colloid, and not the most exciting one in the world. Opals are colloids. There's things called quantum dots that come in all sorts of colors. Some of the oldest colloids were made of gold. That's what stains glass windows. One of the most interesting ones is made by nature, a virus. Viruses are amazing little particles, little machines that can do extraordinary things. Generally, we want to get rid of them, but still you have to tip your hat to nature in what it can do in putting together a particle like this. Turns out that we can't come close to doing something like this. Not yet. What we can do is something like this. So what I show here is an opal. An opal is a bunch of colloidal particles that are put together. On the left of the slide here, there's an electron microscope picture of what's inside a colloid, and what's inside an opal, and it's just a bunch of little colloidal particles. They're arranged together in a nice array, and that nice array is what gives the colloid these pretty colors that you see. We can do this too. We can make little tiny particles like this. We've learned how to do this over the last 50 or 60 years. And basically, if you just confine them, they'll, on their own, assemble into a nice structure, much like oranges stack in a grocer's stand. That's what we can do, all right? We'd like to be able to do more. So this is the sort of structure that you see in the opal. It's called a face-centered cubic structure. Here's another structure. It's a diamond lattice, all right? So diamonds, the gemstone, if you look at the carbon atoms that make them up, they're arranged in this special crystal structure. And the thing that's unique about it is that each particle, each carbon atom in a diamond lattice, is connected to 4 neighbors. It has 2 bonds that go up and 2 bonds that go down. So it turns out that some physicists at Iowa State University predicted that if you could arrange colloidal particles in a diamond lattice, you could make something called a photonic band gap. This would be the optical analog of a semiconductor. Now, semiconductors are the materials that make up all our computer chips that allow us to do these amazing things with electronics. And so the idea here is maybe we could do some of those same amazing things, not with electricity, but with light, if we could just make this colloidal structure into a diamond lattice. Here's the embarrassing part. This was done 24 years ago, in 1990. Nobody's been able to do it yet. We've been working at this for 24 years, and we can't do it. So people on my group, we began thinking about this about 10 years ago, asking ourselves, is there something we can do that can help us learn how to make a diamond lattice? And sort of the goal is to see if we can take these tiny little colloidal particles and attach other particles to them in this special arrangement where you have 2 particles that are up and 2 particles are down, this so-called tetrahedral coordination. So here I just want to sort of summarize in this slide the sorts of things we can make and the associated length scales. So the bar across the picture here goes from 10 to the minus 10 meters, that's the size of an atom, up to a few meters, the size of a car. So we can make things on the size of a car. We can make things that are maybe a centimeter in size. We can even make fine little machines that go in, the fine little parts that go inside of a watch. Chemists can assemble amazing things from atoms. But in this in-between regime, from about a nanometer, 1/1000th of a meter, to a micrometer, 1/1,000,000th of a meter, we're not very good. We can make planar structures. That is, we can make structures in 2 dimensions. But we can't make sophisticated structures in 3 dimensions. And so how do we do this? Well, what we do is, in my lab at least, and in another number of labs across the world, is we use a process called self-assembly, which basically tries to use the physics at small length scales to trick particles, as it were, into assembling into the structures that we would like them to assemble into, all on their own. All right, so what are colloids? Colloids are little particles, roughly 1/100th the diameter of a human hair and smaller. The thing that's, a thing that's very important about colloids is you can make them out of a number of different materials. You can make them out of plastics. You can make them out of inorganic materials like glass and titania. You can make them out of semiconductors for electronics. You can make them out of metals. What would be really great is if we could take those colloids made out of different materials and put them together into structures of our own design. That's something we can't do, but if we could make new kinds of solar cells that were more efficient, new kinds of batteries that are more efficient. We could make 3-dimensional electronics that we can't make at all right now. There are all sorts of things we could make if we could figure out how to put these particles together. So here's our challenge. Make this. So we've sort of taken this diamond lattice as a goal. It's not the only goal. It's maybe not even the most important goal, but it's hard. And so it pushes us to think about how we can develop new ways of putting things together. All right, so here's one thing that we've come up with. We want to make colloids. We say they have valence. By that we mean they bind to a specified number of particles, in this case 4. So what we do is we start out with soapy water. Water plus a surfactant in one jar. In another jar, we put our tiny little colloidal particles with an oil. Then we mix them together, shake them up, and we make little oil droplets inside the water. And inside each oil droplet, if we're careful about how we do this, we can have 1 or 2 or 3 or 4 little colloidal particles. Then we suck out the oil, and when we suck out the oil, we get little clusters of colloids. If there were 4 colloids inside the droplet, we get a little tetrahedron. If there were 2, we get a little dumbbell. If there were 3, we get a little triangle. Now here's actually a picture of the formation of one of these clusters. In this case, there are 7 colloids. They're fuzzy because they're so small, and we're looking at them under a very powerful optical microscope. The droplet they're in is shrinking. It's evaporating into the water around it, and as it does, it confines the colloids into a tighter and tighter shape until at the end, what emerges is this sort of flower-like shape. You can see there are 5 colloidal spheres in a pentagon, 1 above and 1 below. We can see it clearer in an electron microscope picture, but there it is. Every time there are 7 particles inside of a droplet, it makes this structure. So we can catalog them. Here's what you get for 2, 3, 4, 5 through 11 particles. Interestingly enough, this is a side story that I can't go in today. It turns out that mathematicians at Princeton had identified these structures as the structures that minimize the second moment of the mass of these clusters. It turns out that our clusters exactly match those. All right, so like I said, that's a different story that I can't tell you about today. Pictured down in the lower right is the graduate student working in my lab who made these. Now, so what we want to do now is make some patchy particles. So we start and we make this cluster, say this cluster of 4 particles. Then we have a trick that we developed where you swell this cluster with some liquid, and you swell it with just enough so that the tips of each particle stick out of the liquid that you have swollen it with. And then we do a trick. We solidify this droplet. So now we have a solid particle, and the particle has 4 tips of the spheres that it was made from sticking out. Here's what it actually looks like under an electron microscope. So on the top, we have the clusters made up of 1, 2, 3, 4, 5, 6, and 7 particles. You recognize the one in the 7 one as being the flower that we saw before. Below are the particles with the patches that we made from them. And if you look in the middle, you'll see that there's a particle with 3 patches. You see there's a particle with 4 patches, and so forth. So this is a nice thing. Now, it turns out that each of these patches is chemically different from the material that it's embedded in. And what we can do is we can put some glue on these patches, and we can glue DNA to each of these patches. Now, there's an interesting thing about the glue. The glue we use can be different colors. All right, so we like to use green glue and red glue. The reason we do this is so that we know what kind of DNA we've put down. If it's green, we put down one kind of DNA. If it's red, we put down a different kind of DNA. So we did that. The DNA is special. The DNA has a sticky end on the end of it. So what's the sticky end? Well, you may remember that DNA is a double helix. It's made up of 2 strands of molecules that twist around each other. And these 2 strands are connected by molecules called bases. And there's 4 different bases. There are thymine, adenine, guanine, and cytosine. For short, T, A, G, and C. And it turns out when you make a bridge across these 2 strands, the GCs bind to each other and the TAs bind to each other. But the Gs don't bind to the As or the Ts, the Cs, so forth. So what you can do is you can actually get the DNA so that it's double-stranded all the way up to the end, and at the end, it's single-stranded. And you might have on the order of 8 unpaired bases. So here I show a red particle. It has the bases C, A, G, T, G, G, A, T. And the green one has G, T, C, A, C, C, T, A. Those exactly complement the other bases. So you always have either a GC pair or a TA pair. These strands will bind to each other if you lower the temperature below roughly body temperature. And they'll come apart if you raise the temperature above body temperature. So what we have now is we have a way of binding these 2 particles together, but only red particles will bind only to green particles. And the reason is the green particles have the complementary code in the sticky end of the DNA to the red particles. So red won't bind to red, and green won't bind to green. So what we did first is we took particles with 1 patch, which I show on the right in a cartoon AB. And on the left, so we just throw them into a beaker, then we look at them under a microscope. And when we look at them under a microscope, what we see is pairs of particles. Everywhere, pairs of particles. And if we look at the fluorescent glue that we put down, we notice that every pair consists of a red one and a green one. No green greens, no red reds, only red and green. So that means these particles are hooking up by the DNA glue that we put onto them. We can get a little bit more fancy. We can take particles with 2 patches and mix them with particles with 1 patch. In this case, we get colloids that assemble in 3s. And you see again that the middle one has 2 greens, and the outer ones are reds, and you always get these green-red pairs again. We can get more complicated. We can put in one with 3 patches, and then we can attach particles on the end like this. And then this is the one we were really trying to do, 4 patches with the tetrahedral structure. And you see on the left in what's labeled D, a picture of an optical microscope of 4 particles attached to the patches of the central particle. We can do other things. We can put together two patch, green and red particles, and in this case, it forms a linear chain. And if you look at the linear chain, you see that it always consists of red-green pairs. So it's working the way we designed it to work. Here's what it actually looks like under a microscope. So you can see the movies of these particles moving around. The reason they move around like this is because they're being bombarded with the water molecules in which they're suspended, and that results in something called Brownian motion. Now, here's a movie of the formation of one of these AB4 structures. So you see a 4-patch particle and a 1-patch particle. It's hard for you to discern that, but you'll see them hook up right there. All right, so they just, the first one bound to the central particle. Now we'll watch and see that another particle will bind to a different patch on that central particle. So we can actually watch the formation of what we call a colloidal molecule. This is analogous to the molecule methane, which is CH4. You have a central carbon atom that binds to 4 hydrogens. Here we have a central 4-patch particle binding to 4 1-patch particles. And we can watch this sort of chemistry occur in real time. If you looked at this if you could watch a methane molecule form, you'd have to watch very, very quickly because it happens in 1/100 trillionth of a second. All right. So here we have the advantage, and it takes about a half hour. And so we can just watch the formation of this under the microscope, and we can understand quantitatively how it works. You see now we're about to watch the final particle come and attach to that central particle to form this nice tetrahedrally coordinated colloidal molecule. Okay, so there you have it. So we have these new kinds of bonds. Now, what's this all about? You might remember the movie Skyfall. It came out in the fall of 2012. We call this the old bond. All right. Our article describing this research was published in the journal Nature, and the editors at Nature decided to call this the new bond. They came out the same week. All right. I'm going to finish up here, and this is another story, but I'll just show you the results of this. This is something we call lock and key colloids when we publish the articles in the scientific literature. In the lab, we call them Pacman particles. Here's what they look like. All right? They're little particles with a dent in them. And figuring out how to make this dent was the genius of this guy down in the bottom, Stefano Saccana, who's a professor in the chemistry department here at NYU. So I want you to watch this Pacman particle with a little dent in it. It's going to act like a Pac-Man, and you'll see why. Boom. It attaches to that. There's a lot of interesting science behind that. It'll have to wait for another talk to tell you about how that works. But that's the first time that that kind of thing's been done. Now keep watching that Pacman particle, because it turns out it's not quite finished. It finds another one and binds to it. And if you look at it in close, you can see 2 Pacman particles bound to a central sphere. It's a sort of ball and socket joint. It is, I believe, the world's smallest ball and socket joint. No one has ever made one smaller than this. All right. So here is, again, us trying to develop new ways to build up structures from colloidal particles. And in this case, we use this, what's called a depletion interaction. I can't show you everything we do. Here's a whole bunch of micro lenses we've made. These lenses, these lens particles are made out of the same material. It's a plastic. They're about 1/1,000,000th of a meter in diameter. They were made by 2 people, Chasm Edmond and Tess down there. Tess is a high school student working in my lab. She's also in the Juilliard Orchestra. She plays the viola, or no, cello. At any rate, she made these particles, and here's something that Kazem did with these particles. He threw them in water, and they're dancing around by this phenomenon known as Brownian motion, just because they're getting hit by the molecules of water. If we throw in a little bit of polymer, here's what they do. They form little worms, all right? So this, again, is another example of us developing new ways to control how these microscopic particles assemble. All right. So none of this happens by the effort of one person, but by many people. These are some of the people who worked on this. Vinny Manoharan sort of got the colloidal molecules going. Paul Chaikin and Stefano Sakana and I worked on these Pacman particles. The lenses were made by Girai and Stefano Sakana. What all this is to say is this is the work of a big team of scientists working together, trying to figure out how to put these tiny particles together and hopefully build new materials over the next few decades. Thank you.